Thank you very much for the opportunity to present this work with my colleague, Jérôme Ange-Albert, who is here too, and our two PhD students, Maxence Romeran and Jemai Salomon. So I will be talking about our work on quantifying the genetic basis of mixing ability and investigating its genetic architecture with two case studies, one on wheat varietal mixtures and one on wheat-pea intercrops. So first I will start with our motivation based on the agroecological transition. Then I will describe a bit in detail what we mean by the genetic basis of plasticity between stand types. And then I will briefly present the two case studies to estimate covariances mostly and detect QTLs, which is a perspective. And then I will finish with the perspective here. So the main lever of the agroecological transition is to decrease synthetic inputs and to diversify both at the spatial scale and at the temporal scale. And I will focus in this talk on one main practices of interest, which are varietal mixture, the fact to sow several cultivars in the same plot, and intercrops, to sow different species in the same plot. In such diversification practices, we can observe plasticity that is due to plant-plant interactions. So here is a typical image to illustrate phenotypic plasticity, where each symbol represents a genotype, and the two environments, in our case, the first environment will be a monovarietal culture. So sometimes we talk of sole crop or pure stand. And the second environment will be a varietal mixture, but it could also be a crop mixture or varietal mixtures in a crop mixture, etc. And such a plasticity can give rise to, can have a genetic basis, and so we can have crossover interactions, as was already discussed. So this was observed notably in a meta-analysis of cereal legume mixtures, where in 75% of the cases, the interaction between the genotype effect and the cropping system was significant. So this means that monovarietal breeding may be suboptimal when the goal and the target is to produce with mixtures. But obviously there is a big challenge facing breeders, is the combinatorial explosion with large and larger number of genotypes to screen. The number of possible mixtures is bigger and bigger. So one main strategy is to use incomplete design and mix models. So this was proposed, for instance, in varietal mixture by my colleagues. Also applied in the case of intercrops, and it was used as an example with genomic prediction in a breeding scheme for intercropping. But despite all the experimental evidence showing the advantages of mixture, virtually no breeder is using such a strategy. And most breeders mostly work with monovarietals. So it's typical of a lock-in situation where there is a strong interdependencies and self-reinforcing mechanisms within a socio-technical system that is discouraging the stakeholders from adopting an alternative production systems. And so our hypothesis is that we state that proposing a methodology to jointly analyze monovarietal and mixed stands could help breeders to gradually incorporate mixture objectives in their program. So the gradually here is a key point. So for that, we need to formalize precisely what we mean by mixing ability. We need to quantify its genetic variance, assess its stability, identify its underlying traits that determine their genetic architecture, and so on. Okay, so what about the genetic basis of plasticity? So the quantitative genetic framework was worked out by many people. And so here is really a quick overview. In a group of N animals, the phenotype of the focal animal I can be decomposed into a direct breeding value and a non-heritable component, as well as the sum of the social breeding values of the other animal with which the focal animal is living. So the direct breeding value will be the effect of its own genotype on its own phenotype, and the social breeding value of individual J genotype on the other group members. So a key parameter is the correlation between the direct and social breeding value, which can have a big impact on the response to selection. And when this correlation is negative, selection can lead to a negative response. But there are some specificities of plants compared to animals. For instance, they can be easily cloned, and so field data, so as we just saw before, field data are mostly collected at the scale of a group of plants with the same genotype. So here is a typical, so it's a drone picture from a wheat monovarietal stand at 300 plants per square meter, and so usually we collect data at the scale of the microplot. And so when working in such microplots, it's quite important to assess or to develop methodologies to assess genotype proportions in mixture. So the mixing ability in terms of direct breeding value and social breeding value in a varietal mixture sowed in equal proportion, when we only observe the total yield of genotype I mixed with genotype J, we can decompose it in terms of general mixing ability, the GMA, and specific mixing ability of I mixed with J, with the GMA defined as the sum of the direct breeding value plus social breeding value, and the specific mixing ability being 1/2 of the interactions of both interactions. If we have access to the yield per genotype, then we can directly make use of the direct and social breeding values for I mixed with J, and similarly for J mixed with I. But we want to jointly, or to be able to jointly analyze monovarietal, varietal mixtures and intercrops. So we need to distinguish between the social breeding value in a sole crop context and social breeding value in an intercropping context. So all these equations here now, I added the sole crop here, because in a varietal mixture, we only have a single crop. Now if we switch to the crop mixture case, so a binary crop mixture of species S1, so genotype I of species S1 mixed with genotype J of species S2. If we only have access to the total yield, then we can define similarly the general mixing ability in intercropping, which will be the sum of the direct breeding value of this genotype of species 1 plus its social breeding value. And similarly, we can define the specific mixing ability in this intercropping context. And if we have access to the yield per genotype, which is more feasible for intercrops, then we can obtain a similar equation as before. But what about monovarietals? So here comes the second specificity of plants. They do not move, mostly. And so they developed capacity to sense the density of their neighborhood and actively adjusting their development to it. And this is also the case in monovarietals, and it's well known to all breeders, and it has a genetic basis. And so the differences among genotypes in monovarietals for this neighborhood sensing can inform on their behavior in mixtures and reciprocally. So this was proposed by my colleagues in the case of varietal mixtures. You have, so in a binary mixture, you have plant-plant interactions between plants of the different genotypes, genotype blue and genotype red. In each monovarietal, you also have plant-plant interactions. And these plant-plant interactions between plants of the same genotype also can occur in the binary mixture. So they introduced a specific mixing ability for genotype I. So they named it SMAII. It's how the genotype I performs in monovarietal compared to the mean of mixtures comprising it. They extended this model to any mixture orders and genotype proportions, and in terms of plant density, and they showed evidence from field trial in a wheat varietal mixture of positive variance for this specific mixing ability. So, as was discussed in this book chapter, to assess the relative efficiency of indirect selection in monovarietal versus direct selection in mixture, it's crucial to know the genetic correlation between both environments. But of which genetic values are we talking about here? So in a really nice paper that I cited already, they simulated data assuming a correlation between breeding value in sole crop and breeding value in intercrop for these ranges, but they did not express these breeding values in terms of direct breeding value and social breeding value. In analytical studies, they showed the importance of the signs and relative magnitude of the correlation between the direct breeding value and social breeding value, as well as the correlation between the breeding value in sole crop and the direct breeding value. But here again, what is the breeding value in sole crop in presence of specific mixing ability of the genotype on itself? So we can in fact express the classical breeding value in terms of direct and specific breeding value in sole crop. So simply the classical breeding value will be the sum of its GMA plus its SMAII, and the GMA as before is the sum of its direct breeding value and its social breeding value, and the SMA will be the interaction between both. And in some cases, for instance, in field trial with both intercrops and monovarietals, but no varietal mixture, we cannot distinguish between these two terms, and so we can introduce what we call the social intragenotypic value, which will capture the genetic basis of phenotypic plasticity between monovarietal and intercrop due to sole crop plant-plant interactions. So we implemented methods to estimate all these breeding values in a field trial with mixtures and monovarietals, accounting for correlation between direct and social breeding values, as well as between residual errors in mixture, and taking advantage of kinship data. We use for that, so we developed custom C++ code, and we use for that the TMB package implementing automatic differentiation, sparse data structure, and the Laplace approximation to integrate random effects. So it's coming soon. And very quickly, briefly, this is our typical validity domain. We want to be able to analyze field trials of microplots. So we have a first case study in the classical nursery design, and we can show that it's informative to estimate social breeding value, and I encourage you to check out the poster from Maxence Romeran, who will discuss this more in detail. And we also have a case study on wheat-pea mixtures. We have a large trial in Saclay and in Rennes with colleagues with 200 wheat genotypes, in monovarietal as well as mixed with 2 pea testers. So a few weeks ago, we harvested, we sorted the grains between the 2 species, and fresh out of the field, we were able to estimate the variance of the direct breeding value, the variance of the social breeding value in intercropping, as well as the social intragenotypic value, and you see that the sum of both of these terms is not negligible compared to the variance of the direct breeding value. Interestingly, we have a positive correlation between the direct and social breeding value, and we have an error variance of the similar magnitude of the direct breeding value and then with a negative correlation between both species. So I highly recommend to check out the poster from Jimai Salomon. And I finish with a few perspectives, but because I'm quite late, I thank all my collaborators, and I advise you to save the date if you want to hear more about mixing, whether there are crops, varieties, disciplines, or know-hows for agroecology. Please come in Saclay in October 7 to 9 to discuss this in an interdisciplinary setting. Thank you very much.